Hi there, welcome to our IB Biology revision series on section A2.2. So one of the things is in eukaryotic cells is that compartmentalization enables different chemical reactions to occur. So with eukaryotic cells, you've got something called the animal cell, which you traditionally see, and then obviously you've got the plant cell, which is shown in the next slide. So as you can see, the main differences will be aspects such as a cell wall and vacuole. So organelles of the eukaryotic cells include the endoplasmic reticulum, ribosomes, lysosomes, Golgi apparatus, mitochondria, the nucleus, chloroplasts, which are only found in plant and algae cells, centrioles present in eukaryotic cells. Centrioles are not found in plant and fungal cells, and then you've got vacuoles, which are only found in plant cells as well. So the cytoplasm is the plasma membrane outside of the nucleus. The fluid portion of the cytoplasm around the organelles is called the cytosol, and the eukaryotic cytoplasm contains small fibers and rods termed a cytoskeleton. So the cytoskeleton contains actin filaments, intermediate filaments, and microtubules. Microfilaments function in cell division and cell movement. Intermediate filaments shape and anchor some cells. So generally, the cytoskeleton plays a role in maintaining cell shape, anchoring some organelles, aiding cellular movement, and movement for some organelles in the cell. So the endoplasmic reticulum is an extensive network of tubules or channels that exist in all parts of the cell. Its structure enables transportation throughout the internal region of the cell, and there's two types of endoplasmic reticulum. You've got the smooth endoplasmic reticulum, and then you've got the rough endoplasmic reticulum. So you've got the smooth endoplasmic reticulum, and it's very unique. It has enzymes embedded on its surface, and it's involved in a variety of functions. So the production of membrane phospholipids and cellular lipids, the production of sex hormones, the detoxification of drugs in the liver, the storage of calcium ions, which is required for muscle contraction, transportation of lipid-based compounds, and helping the liver release glucose into the bloodstream. So we have the rough endoplasmic reticulum. So these are ribosomes on the exterior of the channels. They participate in protein synthesis, and also this type of endoplasmic reticulum plays a role in protein development and transport. They eventually become part of membranes, enzymes, or messengers between cells. So one of the things about ribosomes is they do not have an exterior membrane and conduct protein synthesis within the confines of the cell. They are free in the cytoplasm or attached to the surface of the endoplasmic reticulum, and one of the things is they're composed of a type of RNA and protein. Now, you've got the differences in ribosomes in eukaryotic cells and prokaryotic cells. So in eukaryotic cells, they consist of two subunits which are equal to 80S, and in prokaryotic cells, they consist of two subunits which are equal to 70S. So lysosomes are basically intracellular digestive centers from Golgi apparatus. So one of the things is lysosomes are vesicles bound by a single membrane, and they have 40 different types of enzymes. They are all hydrolytic and catalyze the breakdown of proteins, nucleic acids, lipids, and carbohydrates. And one of the things is they fuse with old or damaged organelles, so recycling can occur, which is a good benefit for it as well. They're also involved in the breakdown of material brought into the cell by phagocytosis. And one of the key things is that the internal environment of the lysosomes is acidic. So the Golgi apparatus consists of flattened sacs called cystinae, and the key function includes packaging, collection, modification, and the distribution of the material in the cell. Now, one side of the Golgi apparatus is called the cyst side, and then the products move from the cystinae of the Golgi apparatus, and then they basically move out to the trans side. Now, one thing is that lysosomes are important examples of vesicles and carry material inside and outside of the cell. So the mitochondria is a very important organelle. They basically appear throughout the cytoplasm. They have their own DNA. They have a double membrane. So they have an outer membrane that is smooth and an inner membrane that is folded into cristae. The inner membrane is basically a semi-fluid substance called the matrix. And one of the things about the cristae is they have a large surface area where chemical reactions occur, and they're involved in the production of energy called ATP. And one of the other things that you may hear about the mitochondria and reactions that go on is things like glycolysis, the link reaction, and this will be covered in a lot more detail. So this will be covered in section C 1.2. So the nucleus is basically referred to as the genetic powerhouse of the cell. And one of the things is that the nucleus contains a double membrane, which is referred to as the nuclear envelope. It allows the compartmentalization of DNA. Now, DNA is found in the form of chromosomes, and this consists of all the kind of genetic information. So when a cell is not dividing, chromosomes are not visible, and then DNA exists in a form of chromatin. Chromatin is formed of strands of DNA and proteins called histones, and DNA and histone combination results in a structure called a nucleosome. And on the right-hand side, you can see what the structure of the nucleus looks like. So chloroplasts, these occur in algae and plant cells. Now, one of the things is the chloroplasts again have a double membrane, and it has its own DNA and a 70S ribosome, which is important to remember. Now, the interior of a chloroplast includes grana, thalcaloids, and the stroma. Now, with the thalcaloids, they are flattened membrane sacs, and these contain the components for the absorption of light. Now, as we know, photosynthesis is a chemical process. It converts light energy into chemical energy. Now, the stroma contains many enzymes and chemicals necessary to complete the process of photosynthesis. And in section C 1.3, we will go over some of the things about the photosystem 1, photosystem 2 in greater detail. So the centrosome occurs in all eukaryotic cells. In animal cells, a pair of centrioles are involved in the assembly of microtubules, and they provide structure and allow movement. So, vacuoles is a storage organelle which is formed from the Golgi apparatus. In plant cells, they have a large space. Now, vacuoles consist of many different substances, including metabolic waste, toxins, and water. And one of the key things is it enables cells to have a higher surface area to volume ratio. So generally, one of the comparisons you have to look at is the difference between prokaryotic cells and eukaryotic cells. So with prokaryotic cells, DNA is in a ring form without a protein. In eukaryotic cells, DNA is with proteins and obviously chromosomes and chromatin. DNA is free in the cytoplasm. DNA is enclosed with a nuclear envelope in the eukaryotic cells. Prokaryotic cells have no mitochondria. Eukaryotic cells have mitochondria present. And then you've got the difference between the 70S ribosome in prokaryotic cells and the 80S ribosome in eukaryotic cells. Another key difference is the compartmentalization. So within prokaryotic cells, there's no internal compartmentalization. And with eukaryotic cells, there is internal compartmentalization, and then that allows many types of organelles to be formed. So prokaryotic cells are less than 10 micrometers, and eukaryotic cells are more than 10 micrometers. Thank you very much for listening to this presentation on section A2.2. We will be doing more recordings in our IB Biology revision series.